Right. Good afternoon, everybody, and welcome to the Genomics England Research Seminar for May this year. We've got a really exciting seminar coming up with two groups presenting, both reporting about a non-coding variant in the gene RNU4-2 as a cause of neurodevelopmental disorders, both making discoveries out of the National Genomics Research Library to achieve this. The first presentation will be by Yuyang Chen from working with Nikki Whiffen, and the second from Daniel Greene working from Ernest Turo. So we'll go to those shortly, but just to, at the beginning, just highlight that this seminar is held at 2 o'clock on the last Tuesday of each month, and the next seminar is going to be from Professor Raj Thacker from Nuffield Department of Medicine at University of Oxford, who's also going to be talking about genomics of neurodevelopmental disorders, and that will be followed by a presentation from Genomics England's program talking about our long read and multimodal program. So without taking up any further time, I will pass over to Yuyang to present about patients in neurodevelopmental disorders. And just to remind people that if we can put questions in the Q&A box that you'll see down the bottom of your Zoom screen, we'll get to questions at the end. And if we don't get through all of the questions in the discussion time, then Yuyang and Nikki have agreed to stay on and provide written answers to the questions we don't get to. So without any more ado, over to Yuyang to present. Thank you.
Thank you very much, Matt. I'm just going to share my slides now, and then I'll start my presentation. Okay, is that everything?
That's clear, yes.
Yep. Right, great. Thank you. So I'll start now. Okay. Hello, everyone. I'm Yuyang, and I'm a DPhil student at the Computational Rare Disease Genomics Group in Big Data Institute, University of Oxford. And today I'm very excited to share with you our group's finding on de novo variants in the non-coding snRNA gene RNU4-2, frequently as a frequent cause of neurodevelopmental disorders. To start with, I'm going to introduce some basics on neurodevelopmental disorders, or NDD. So NDD are a group of conditions with onset in the developmental period. And the disorders typically manifest early in development and before the child enters school. And they are usually characterized by developmental deficits that impairs personal, social, academic, or occupational functioning. So currently, using current diagnosis approaches, we can achieve around a 40% genetic diagnosis rate in recent efforts. So this is from the Deciphering Developmental Disorders studies, and it shows that around 40% of NDD individuals can have a genetic diagnosis at the moment. And our group's interest is to identify new genetic diagnoses for these individuals, primarily in the non-coding regions, which has largely not been a focus of previous genetic diagnosis approaches. And the Genomics England datasets we're really lucky to use. It contains a lot of whole genome sequencing data, and also of a large cohort of NDD individuals, giving us a lot of power to identify such new diagnoses in non-coding regions. So to identify new diagnoses in these non-coding regions, we looked for de novo variants that are enriched in non-coding regions in the GEL NDD cohort. So this cohort is defined by considering the disease that each of these participants were recruited for, and also the phenotype records of each of those participants. So in the GEL high quality de novo core set, we were able to identify 3 cohorts, the first being diagnosed NDD, which are participants with NDD phenotypes that has been reached a genetic diagnosis. And we also have of interest undiagnosed NDD participants, which are 5,426. And we also have participants without phenotypes that are matching NDD standards. So this is about 4,776 participants. So within the undiagnosed NDD cohort, I identified a striking single base insertion on chromosome 12 in the gene RNU42 while investigating these de novo variants in non-coding RNAs. So there are a total of 34 individuals that we found with this variant in the undiagnosed NDD proband, and 46 found in total in the entire 100K genomes cohort within GEL. And this variant is also not found in individuals without NDD phenotypes or diagnosed NDD phenotypes, and is also only found in a single individual in the UK Biobank, which is a large cohort of about 500,000 genomes, meaning that this variant is incredibly rare in the general population. Given that so many individuals have been found with this variant, we hope to confirm that the variant is genuinely present in these individuals. So to do this, we looked into the quality control metrics of these samples and compared that with other samples, and also we compare the region of RNU42 with other regions in the same samples. So when we compare the quality control metrics of the individuals with RNU42 variant, we found that there is no significant difference in terms of genotype quality score, and also sequencing depth for if we compare the participants with the variant to the participants or the other participants in the AgV2 aggregated variant set within GEL. So there's no significant difference. And within these participants with the insertion variant, highly recurrent insertion variant, we also compare the coverage, in this case measured by the number of reads that has mapping quality greater than 20 of RNU42 and of other regions of the same length, one of which being RNU41, which is a homolog of RNU42 that's just nearby RNU42, and also 999 other intergenic regions that are selected at random of the same length of RNU42. So in this case, we also did not find any significant difference in terms of the coverage in these regions. In addition to that, we have also confirmed the variant's presence in 5 individuals and their absence in their parents with Sanger sequencing. In addition to that, out of these 5 families, in 3 of them, we have both long and short read sequencing to confirm the presence of this variant. So we are pretty sure that this variant is genuinely present in these individuals. Given the high recurrence of this variant, we sought to identify if there is any other variants of interest in the region nearby or on the RNU42 gene in general. So in this case, we have found a depleted region in the UK Biobank population cohort of 18 base pair. So this is a very lack of variants within the UK Biobank, with only a very few variants found, whereas it is highly enriched in variants, especially insertion variants of this highly recurrent variant in the GEL NDD cohort. And in fact, if we calculate if we use simple burden test to calculate the burden of this variant, we can see that the odds ratio of insertion variants in this critical region has 1,000 higher than 1,500. So there's only 2 insertion or deletion variants found in the UK Biobank in this region, compared to 55 in GEL NDD cohort. And we also find a more modest enrichment of single nucleotide variants within the GEL NDD cohort as well within this region, but this is much less pronounced than insertion variants. We have also looked into additional cohorts to see if there are any other individuals with the same variants or variants in the same regions with NDD, and we have been able to identify some more individuals in this critical region, and the situation is similar to what we have seen in the GEL cohort, with many of them found with the highly recurrent 6465 insertion T variant and some other variants as well. And many of them have also been confirmed to be of a de novo origin. In total, we have found 116 NDD cases within this critical region, of which 90 of them being the highly prevalent insertion variant. So given that so many individuals have been found with this variant, we hope to characterize fully the phenotype of the individuals with this variant in this critical region. So to date, we have been able to collect phenotype data from 50 of these individuals with the variant, and we are seeing that RU42 variants are associated with moderate to severe NDD phenotypes. So of the phenotype information we have collected, we see that all the individuals are affected by global developmental delay and intellectual disability, and 97% of them are affected by speech abnormality, 97% hypertonia, and 97% of them have dysmorphic facial features. And there are also other NDD phenotypes affecting them as well. So around 76% of them have seizure, 80% of them have microcephaly. And this variant also affects other systems of the body as well. So about 88% of them have vision issues, 87% bone or skeletal issues, and 86% short stature. So I'm just going to show some common facial features, so some patient photos in individuals with RU42 variants. And so in these patients, we can see that clearly there's features of myopathic face, deeply set eyes, and a distinctive mouth, full lips, and downturned corners, and a larger protruding tongue. So this has been reviewed by numerous clinicians. who agree that there is a significantly very common facial features shared between these participants. Given that so many participants are affected with a similar NDD phenotype, we hope to understand the underlying mechanism of RU42 causing these kind of NDD phenotype. And according to genome annotations and our understanding of the RU42 gene, it is annotated as an snRNA, it's a small nuclear RNA of the major spliceosome. So this is highlighted here in pink, so it codes for the U4 subunits of U4 snRNA of the major spliceosome. And it pairs up with the U6 to form a duplex and catalyze ensure that 5' splice sites interaction are correctly recognized. So if we look more closely into the interaction between the U4 and U6 snRNA, we can see that there are some critical regions of these interactions. So notably, we have stem 1, in which U4 and U6 base pairs, and also stem 3, there's also some base pairing in here. And in the middle of stem 1 and stem 3, there's the Akagaga sequence on the U6, and this is essential for correct recognition and binding of the 5' splice site. And also on the U4 of this part, it's called the quasi-feudal knot or the T-loop structure. And the variant we are finding falls in stem 3 and this T-loop structure of the variant. And we think that insertions in this region or some SMVs might disrupt base pairing in stem 3. And also might disrupt the correct recognition of the Akagaga sequence to the 5' splice sites correctly. So we tested this hypothesis, and we found that RU42 variants indeed result in systematic disruption to the 5' splice site usage. So we have 5 blood samples of individuals with RU42 variants, and we found that they have significantly higher usage of annotated or abnormal 5' splice sites in these samples. So as you can see in this panel, in the first panel, we have cases. So we use Fraser to detect abnormal splicing events in cases. So in these 5 cases, we see a significant increase of these abnormal splicing events compared to 378 controls and 10 match controls with cases. So 2 match controls for each case based on sex and age, and also inferred genetic ancestry. And this second panel, we can see that individuals with RU42 variants on average share 11 events between at least 2 of their samples. Whereas in general, in individuals without RU42 variants, we're not expecting to see any incorrect splicing events shared between samples. So this also shows that RU42 variants result in a systematic disruption in 5' splice site usage. So given that RNU42 has so many different variants that could explain NDD phenotype by affecting splicing, and also we know that there are two other snRNA genes that causes rare diseases in a recessive manner. We wonder if other snRNAs would harbor new diagnoses for NDD participants as well. So we examined 28 snRNA genes that we found to be expressed in the brain. But we did not find any burden across any of them except for RNU42, and there are no other variants that are enriched in a de novo manner. And since we have identified an 18 base pair critical region that harbors most of our variants in RNU42, we thought of the possibility of the specific regions having an impact of having variants, for example, having variants enriched in NDD individuals. So we devised an approach to assess the region depletion, the depletion of variants within specific regions by using an 18 base pair sliding window on each of those snRNAs. So this figure describes a sliding window approach to identify regions that are depleted of possible single nucleotide variants. And we see that multiple snRNAs, such as RNU41, RNU5B1, RNU12, etc., and RNU42, notably, have regions depleted of variants in the general population. However, apart from RNU42, we still did not see any other regions that are enriched in de novo variants in the undiagnosed NDD cohort. And very notably, we can see that in RNU41, which is a homolog of RNU42, also coding for the U4 snRNA, has a depleted region, but there are no de novo variants that are significantly enriched in the NDD cohort. We went on to ask why we see variants related to causing NDD phenotypes in RNU42, but not in RNU41. And we investigated this question by looking into the RNU42 and RNU41 expression in the brain. And in brain samples we found that RNU42 is expressed in a much higher level than RNU41 in the brain. So on the panel on the left, you can see that across different developmental stages, RNU42 is consistently more highly expressed than RNU41, which is in gray in here. And also using ATAC-C assay data, we can see that RNU42 is has a much higher chromatin accessibility compared to RNU41. So these all suggest that RNU42 is highly expressed and can affect splicing in the brain, thus leading to NDD phenotypes. So culminating all these findings together, we have published a preprint. So you can find more information in the preprint if you're interested. And we are delighted that we have been in contact with so many clinicians who agreed to provide data of patients that helps us fully characterize this cohort. And since the publishing of this preprint, we have been extensively engaged in public efforts to promote this finding to clinicians, the patients, and to the public. So the first part of it is to deliver diagnoses to patients. So we have collated data from a large number of different cohorts in clinical centers in multiple countries. So this includes the Gregory Consortium, Undiagnosed Disease Network, Sun and Simplex Collection, Matchmaker Exchange, and the NHS Genomic Medicine Service. And this has allowed us to send this diagnosis to many of the patients. And on the GEL side, we have sent more than 50 clinical collaboration requests to inform clinicians of the discovery and to collect phenotype data. And on the patient engagement side, we have also been directing them to Unique, which is a charity supporting individuals with rare chromosome and gene disorders. And we are also aware of a Facebook group of patient families connecting together. And we have been contacting clinicians to direct patients to this group if they're interested. And our preprint has also sparked public discussion on, for example, whether we can identify such variants in exomes. So two scientists on Twitter has shared their approaches to try to identify these variants in exomes. And one of them has some success in identifying some reads of RNU42 with a variant in exomes, but usually the read number is very low because the reads come from a mismapped read because RNU42 is not usually included as it is a non-coding gene. But that shows on one hand, that shows the level of public engagement we have had so far since the publish of our study, and also it also shows the limitations of exome sequencing and the significance of genome sequencing in this case of diagnosing individuals. And there are 2 points I want to kind of discuss before going into the conclusion. So there's one thing we also wonder. The one thing we wonder is why this variant is not found before. So the first thought is that the variant is outside the protein coding region, so it's not been a target of many diagnosis approaches we've had at the moment, so exomes, panels, for example. And also there are very few tools to predict the impact of variants on non-coding RNA. So it's really hard for us to know unless we specifically look into it with a large amount of effort. And also the variants in this region are mostly de novo, which means that we cannot use approaches that's related to inheritance to identify variants that are causing disease in this gene. And another thought from us is that why this variant is so recurrent. So because this variant is one of the most recurrent variants we have found associated to be with NDZ, we thought of 3 potential reasons that it might be recurrent. So firstly, germline selection is possible. So this variant might confer an advantage in the germline, so it might be selected for. And another reason might be a high mutation rate in snRNAs. So especially RNU42 and 41. So in the UK Biobank, we are seeing more than 75% of all possible single nucleotide variants in RNU42 and RNU41 compared to a relatively low proportion in random intergenic regions. So these snRNAs are naturally subject to high mutation rates. Of course, that does not explain the fact that there's only one single recurrent variant on its own. So we think that the local secondary structure might also drive the higher mutation rates, thus creating this very recurrent variant. So in conclusion, we have identified variants in the 18 base pair region in RNU42, which primarily consists of single base insertions, causing around 0.4% of NDD cases. So this considers the upstream diagnosis rate of 40%, and also considers the entire size of the gel NDD cohort and the number of individuals found with this variant found in this cohort. So this makes it one of the most common genes to cause NDD identified so far, compared to many other protein coding genes that's been identified before. So apart from that, we see that such variants in RNU42 result in moderate to severe NDD phenotypes by creating abnormal splicing events. And this really highlights the significance of whole genome sequencing in rare disease diagnosis. And since our finding, we have worked proactively to promote them and also to deliver a diagnosis to clinicians and patients to help them. So this concludes my talk, and I would really like to thank a lot of people who has contributed to this work. So thank to my supervisor, Nikki Whiffen, who has been unreservedly supporting this work and collaborating with a lot of people to put this work together. And also our group's members, Hyunchu, Ruby, Alex, and Elston. And also our collaborators from the Broad Institute and from the IDRM in Oxford and from Genomics England. And in particular, we would like to thank all the participants, clinicians, and recruiting centers for making this possible. Of course, we would like to also thank Genomics England to make this research possible through access to data and findings in the National Genomic Research Library. Thank you very much for listening. I hope you've enjoyed, and I'm happy to take any questions.
So beautifully presented, Yuyang. Thank you. There are stacks of questions, and this is very pleasing because this means you don't have to suffer any from me. So first up, one that was pretty a self-evident question: the one patient from the UK Biobank who carried the variant.
Yeah.
What's Did they have a condition, or were they
Okay,
actually
so
fully healthy?
from the data available we have, so we can have like ICD-10 codes for this participant in the UK Biobank, and we see that the participant has a history of nervous system diseases, and that is pretty much the extent we can get regarding the data we have. And of course, the participant also have other phenotypes as well. So we are unsure, but we do see something related. Yeah.
Is it possible through UK Biobank to go back and actually ask for more clinical data about individuals?
I'm not sure.
No.
I probably I'll probably need to ask some of my teammates because I usually specialize in the gel environment, whereas they kind of do some.
I don't think it is.
Yeah.
Probably.
Okay. All right. Okay. So look, then a couple of people, Charlie Rowlands and an anonymous attendee, have asked the same question about variants in other snRNA genes, and that you mentioned about whether they have phenotypes in other types of diseases other than neurodevelopmental disorders. Is this purely a condition which affects or mutations in these genes purely affect CNS development, or do they affect other systems as well?
So from what we see in RNU42, there is primarily an NDD, so neurodevelopmental phenotype, but we also see other aspects of the individual's development. And in our kind of burden analysis of snRNAs, we have only done the NDD cohort, so we're kind of focusing on neurodevelopmental disorders at the moment. But we think it definitely is possible that there can be other diseases associated with such kind of snRNAs. Yeah, but we are kind of having a focus on neurodevelopmental disorders, and because we're also focusing on snRNAs that are expressed in the brain as well.
Okay, all right. Do you have a hypothesis as to why these are autosomal dominant, and the comments made that other condition affecting the spliceosome typically autosomal recessive?
So we see there are two other genes, so namely RNU4 ATAC and RNU12. So these two genes have a recessive mode. So also variants of these genes are associated with disease, but in a recessive manner. So so I think there are evidence from both sides. Yeah, we've also been looking into variants, and for example, homozygous variants that has inherited one copy from each of the parents, to see if there's any kind of recessive kind of enrichment of recessive variants or compound heterozygotes in these snRNA genes, but we also don't find anything additionally significant.
Okay, all right, cool. So there's questions from Eduardo Calpina and Andrew Wilkie, both asking about what's the replication mechanism that leads to the 3/4 of them being insertions of T. Do you have an explanation as to why that's the case?
So from what I know, the snRNAs, because they are very highly expressed, as they are very important in the biological processes of the cell, so they are replicated by a different DNA polymerase, so DNA pol 3, and that has a much higher mutation rate than the usual DNA polymerase. So that's what I know.
Okay. And so could I ask a question about the evolutionary conservation of this particular gene? Is it one that's actually preserved going a way back, or is it a more recent evolved gene?
I think I haven't actually looked at it yet.
Perhaps I can answer that one. Yeah, so we know it's perfectly conserved in mice. We know it doesn't go back a huge amount further than that.
Okay. So interesting that it should be so critical to neurodevelopmental disorders and yet not actually be preserved.
Yeah, it's also on the point of recessive versus dominant. I think it's neat to note that the recessive genes are both components of the minor spliceosome, which is only responsible for splicing a subset of about 300 introns, whereas this is dominant and the major spliceosome, so that maybe explains the recessive versus dominant effect. So
maybe less tolerated because that spliceosome has a greater functional role.
Yeah, exactly.
Okay. All right, look, we've reached the half-hour mark. Yuyang, thank you so much. Excellent presentation, excellent response to the questions.
Right.
Okay, so I will now pass to our next speaker, who's Daniel Greene. And so, Daniel, if you could join us on screen. Daniel, welcome to the Research Symposium seminar series, and I'll hand over to you to present also on the topic of RNU4-2 variants in neurodevelopmental disorders. And Daniel, just as you missed the briefing beforehand, just to note that after 20 minutes, we'll take questions, which we'll do through the Q&A section at the bottom, and I will moderate them for you. Okay, great. Over to you, Daniel.
Okie dokie. Slides. Can everybody completely
Yes, we can see that. Yep, that's fine.
Okay, great. So the title probably sounds quite familiar to you. And just to say congrats to you, Yuyang, on a great talk. My name is Daniel Greene. I work at Icahn School of Medicine in New York, and I'm a visiting scientist at University of Cambridge. So it's my pleasure to talk to you today about some recent work we've done. Mutations in the U4 sRNA gene RNU42 cause one of the most prevalent monogenic neurodevelopmental disorders. So this work is the outcome of a broader project that we've done, which is essentially etiological discovery in the 100 kgp. This diagram on the right shows a kind of overview of our general operating procedure, where we start by selecting variants. We run within a rare disease sequencing study. We run a systematic cohort-wide statistical association. We then prioritize results, do functional follow-up studies, and then feed this back into the diagnostic process and clinical reporting. So hopefully increasing diagnostic yield. So here we've obviously done it in the 100 kgp. The starting point for the analysis was a merged VCF of 77,000 participants, in which we selected rare variants. We determined, it contains 30,000 probands, 5,000 other affected relatives, and the rest unaffected relatives. We determine case sets based on disease classifications provided by the 100,000 genomes. And we use our statistical association method, BeviMed, which gives you a Bayesian model comparison between model of no association with ones for dominant and recessive association, and enables you to infer a posterior probability of association between the gene and a given case label, but also conditional on the association individual variant level probabilities of pathogenicity. We then take the strongest signals from this and acquire additional orthogonal evidence. We might be looking in more detail at the phenotype or seeking replication of people who have the same or similar variants in collections where there are phenotypically similar cases. And then we would go to validate these discoveries experimentally, collaborating with other groups. So we set the case sets based on the specific disease classifications given in the 100,000 genomes. So these classifications are hierarchical. So specific diseases are grouped into disease subgroups, which is grouped into disease groups, in order to allow for a degree of phenotypic heterogeneity in the association. We treat disease subgroups as case sets in addition to specific diseases. So this panel on the left gives you a breakdown in disease group of the size of the different case sets that we're analyzing. So these are arranged by the size, the number of probands. We've got disease subgroups in blue and specific diseases in red. On the right, we're just zooming in on the neurology and neurodevelopmental disorders. So you can see the top 4 subgroups by prevalence, and then within neurodevelopmental disorders, you can see that by far the biggest specific disease by number of probands is intellectual disability, 5,529. So we did a statistical analysis this was work from 2021, where we looked at all of the coding and non-coding genes for each disease classification. So there were 269 case sets which we analyzed in total, following the procedure outlined in the previous slide. So this contained 20,000 coding, 40,000 non-coding genes. We then selected the the associations for which we inferred a posterior probability of at least 0.95, and we annotated them with PanelApp by matching panels to case sets, and we found that 241 were previously labeled or known, and there were 19 unlabeled associations amongst the coding genes. We also found associations with non-coding genes, including RNU42, and so we started by looking at the coding genes before going to the second chapter, which is what we're looking at now, the non-coding results. And so I'll just briefly describe the outcome of the coding genes analysis, which we published last year. So here you can see on the these little sections contain the associations between genes and diseases that we inferred, with posterior probability of association on the y-axis, arranged by the disease groups. And the colors of the points indicate the evidence levels in PanelApp. So we go from the green high evidence down to red low evidence, and then it's gray if they were unknown. So in this paper, we validated 3, and then also since then another one has been validated. So looking now at the association results for intellectual disability, we found, and the non-coding genes, we found only one gene with a posterior probability above 0.5, which is RNU42. We inferred a posterior probability of 1, so obviously there's very strong evidence, and there were 3 variants for which we inferred a posterior probability of pathogenicity, which was greater than 0.5. The highest probability being this position 64 insertion. So we looked at these 34 intellectual disability cases who carried one of these 3 implicated variants, and we noticed that there was a depletion of cases who had been already explained with an LP, likely pathogenic, or P pathogenic variant. So across intellectual disability cases in 100,000 genomes in general, this is 20% are explained. And so using a one-sided binomial test, we found a p-value of 4.86 times 10 to the minus 4. Of adding further evidence, this was a causal association. We then looked at the HPO phenotypes of the 34 cases with an implicated variant. We defined the phenotypic homogeneity of a group of cases to be the mean semantic similarity between pairs of cases in the group, which we used, we based on Resnick's measure of similarity between terms. We then compared this under a one-sided permutation test to the distribution of the phenotypic homogeneity of randomly selected sets of 34 individuals from the group of intellectual disability cases in GEL as a whole, about 5,000 people, and we found that nearly all of these randomly selected sets had a lower phenotypic homogeneity, and this gave us a p-value of 2.33 times 10 to the minus 5. So again, an additional source of orthogonal evidence for the association. Then we looked at looking at the HPO and the HPO codes of other people with these variants. We noticed that there was significant phenotypic overlap. So people shared HPO terms such as seizures and morphological brain abnormalities. So we expanded in order to capture as many cases as possible, we expanded the definition of the affectedness to include any neurodevelopmental HBO terms or ICD-10 codes. Then by looking out to, by expanding the regions that we are looking for variants, we noticed nearby there were adjacent variants. People had also very similar phenotypes. And these regions, if you looked up until there were any unaffected people who had a rare variant, they form two critical regions, which are highlighted in yellow in the diagram below, which we'll talk about in a second. So by doing this, by expanding this set of variants and definition of the phenotype, we observe 47, we increased the total number of suspected affected cases to 47. And then based on the parental genotype data that's available, we could say that 37 of them were observed de novo, and 10 of these 47 people we don't know. So in this diagram, we can see each base of the RNU42 cDNA, which is 141 bases long on the seq track underneath the bar chart, and then the number of carriers within the 100 kgp merge VCF who carried a rare allele, partitioned by the affection status, where they have an NDA code or NDA ICD-10 code, and also the inheritance status of that variant. Then the track underneath the sequence, the NAA, shows the number of alternate alleles observed at that position. And then right at the bottom, we've got this track GAD, which is just has a filled in circle for where the variant is observed in Nomad. And so you can see that there's this sort of depletion of little black dots underneath the highlighted region. And obviously this is consistent with purifying selection. And the other thing to note is, of course, that there is this 64 in base 64 insertion, which is clearly the main variant with the highest number of cases. So then just to look at the quality of these variants and do a mosaicism analysis, we looked for each variant at the distribution of the number of reads supporting the alternate allele for each of these variants observed in the critical regions in 100 kgp. As you can see, so here the affected heterozygotes are shown in red, and other cases are shown in gray. And you can see that in all cases, the affected heterozygotes have at least 9 reads supporting the variant, and other cases have at most 3, with the single exception of the 65A to G, where the unaffected mother of one of the heterozygous probands for this variant has 8 reads. So this is, you can see that in the diagram, you can the pile up of reads where the gray reads show the reads that support the reference allele, and the blue reads the ones supporting the alternate allele. So this is why we suspect that the unaffected mother is a mosaic. So we then replicated this finding in 3 additional cohorts, which included the NIHR Bioresource Rare Diseases study, the GEL pilot study, and the NHS Genomic Medicine Service. So in this diagram, we see the family structures where the data was available for all of these 47 people from the 100,000 genomes, which was the discovery cohort. The labels are shown in black, and 27 additional families who were found in the replication cohorts, which are shown with blue labels. As you can, so clearly there's the same pattern of observing the 64 insertion being the most common variant out of these. Also notably, there was one instance of inheritance. So in pedigree S21, there was an affected mother and an affected child who had this base 76C to T variant, which is also the only variant which is observed in Nomad. And clearly is, we suspect it to be causal because it's also observed de novo, for example, in S20 and S22. And there was an unaffected case who carried a different variant not observed in the 100 kgp, which was the 76C to G variant. So suggesting that the penetrance of variants at this position is lower than for at least the predominant variant, the 64 insertion. So this diagram shows the encapsulates the characteristic phenotype of the RNU42 disorder. So here we're showing the 46 NDA, so neurodevelopmental abnormality HBO coded cases from the 100,000 genomes. There's one case who erroneously didn't have an NDA HBO code, but they did have ICD-10 codes, which revealed that they were indeed affected with neurodevelopmental disorders. In this diagram, we're selecting terms which are present either in at least 50% of the 46 cases, or which are significantly enriched in comparison with the other people who have neurodevelopmental abnormalities in Genomics England. So that's 9,112 people. So the significantly enriched terms are shown in yellow, with the little red arrow, the base of the arrow being the prevalence of that term in the non-RNU42 carrying NDA-coded part of the cohort, and then the head of the arrow being the proportion amongst the 46 carriers. When the number of people is shown on the term with the percentage amongst the 46 as a percentage in brackets. So just to pick out a couple of examples, hypertonia, microcephaly, short stature, seizure, and drooling are some of the enriched phenotypes for this disease. So we then looked at prevalence of the RNU42 disease, both in the 100,000 Genomes Project and the Genomic Medicine Service. So in the 100,000 Genomes Project, it was the most frequent cause of, so this is just looking at the NDA-coded participants in each cohort. And for the 100,000 genomes, it was the most prevalent, with 46 of these people explained, and with ANCAR D11 being second. So because the 100,000 Genomes Project, because the Genomic Medicine Service isn't pre-screening for aneuploidy, like Down syndrome or copy number variants, we thought it might give a better estimate of the true prevalence. So in the GMS, we found it was the second highest. It was second only to MECP2, the cause of Rett syndrome, and there were 21 explained NDA-coded cases, which accounted for 0.38% of the whole cohort. So in this diagram, we can see the secondary structure of the U4 sRNA and the U6 sRNA within the U4, U6, U5 tri-SNP. The critical regions containing the pathogenic variants are highlighted. And as you can see, they correspond to binding regions between the two RNAs in the stem 3 and so-called quasi-pseudo-knot of the U4 sRNA. The critical regions are obviously adjacent to the ACA-GAGA loop of U6, which interacts with the 5' splice site of the target intron, an interaction that is vital for activation of the spliceosome. So the implicated variants may disrupt these bonds, and therefore the splicing activation this is going to require further mechanistic exploration. So I'd just like to conclude there and give out my thanks to all the people who've been involved with this and helped support the work, in particular Professor Ernest Thurow and Professor Andrew Mumford and Professor Kathleen Frisson. Also Chantal Thiss, Els Orbitus, Ian Berry, Joanna Jarvis, who've been instrumental with acquiring data for this publication. Also Peter O'Donovan from Genomics England, clearly works late helping with the airlock requests on lots of occasions. And then our funders and of course NHS patients and participants, the 100,000 Genomes Project, who've made this possible, and Genomics England for making this data available. So thank you very much for listening.
So Daniel, another fantastic presentation. Thank you so much. Some really fascinating biology as well as the genomics behind it all. There are some questions in the chat, but please feel free to add more people in the audience. I'm going to kick off with a question which I think most of us are probably sitting here thinking, how was it the disease that common, that's essentially monogenic, which had a distinct phenotype, as you demonstrated, which is, you know, probably the second most common cause of neurodevelopmental disorders, was missed all of this period of time? It's not been critical of clinicians at all. It's just that I think it's really fascinating to look in retrospect and what lessons there might be for other disorders about how this was almost in plain sight and not picked up.
I mean, I think that's a great question. It being a non-coding gene, I know that some people might be suspicious, seeing the prevalence of the variant might make people suspicious. And I know at least one of my colleagues would say that, you know, if they see the same de novo variant too many times, they'll assume it's some kind of artifact or contamination. Yeah. So they won't necessarily look at it. But I would also like to understand that a little bit better, how it was missed for so long. And
so do you think this is something we should be worried about us missing other non-coding variants that are common causes of monogenic diseases?
I think it highlights the importance of looking for this phenomenon, I suppose, to see the same variant, to see such a recurrent de novo variant and to be systematic about it. So it's a good, I guess, a good wake-up call in that regard. But yeah, I suppose it's a sign that we should make sure that these aren't being missed. But
all right. So related to that, you mentioned that there was a high prevalence of skeletal morphology changes, and given that we've got some expert skeletal morphologists on the line, like Andrew Wilkie and co, you mentioned short stature. Apart from that, were there any other characteristic skeletal morphology changes?
There were the morphological brain terms.
No, that axial skeletal morphology and skeletal morphology.
Skeletal, I'm not sure about perhaps at some lower frequencies amongst those cases or for specific variants. Yeah, I'm not sure.
All right. Okay, cool. So I'm going to repurpose a question from Alastair Pagnamenta, who was actually asking Yuyang, but I'm wondering, did you have a look at the RNA sequencing data that was available for these individuals, and retrospectively, could you pick it up from that? Do you think that, given that we are now extensively using RNA sequencing in these cohorts, that we might be able to pick up on diagnosed cases through that?
I would. Oh, sorry. Would you like to take that, Yuyang?
Yeah, I mean, like, just some thoughts on it, because like we do see RNA data, RNA-seq data, and if we look into abnormal splicing events, we might find it, but But the thing is, like, I think that there might be other diseases as well that can cause this kind of abnormalities in splicing. So I think you probably need a very integrated approach. You probably want to see like phenotypes, and you can do, like, if you suspect that, you can probably do a targeted sequencing, which is really easy to do and cheap as well. So yeah, so you can do like multiple approaches to diagnose.
All right, and related to that, Danielle Humphreys asks about methylation patterns, and given that some of these had long read sequencing done, have you looked at the epigenetic change, and is there epigenetic change that we should pick up?
I pass. I haven't seen the long read sequencing data. I wasn't aware about it. But it would be very, very interesting to look at.
Okay, so there are a couple of people who've asked about functional work to confirm the diagnoses. So the question is, have you done any?
We have some. We have some.
Or is it purely in silico?
Work. Yeah. We have functional work underway. Yeah.
Okay. Another person's asked about a facial phenotyping tool and whether it shows phenotypic resemblance. In other words, whether, I guess, we can pick up using facial recognition tools the diagnosis. Is it classical enough for that?
I hadn't seen any facial phenotyping before Yuyang's talk, but I think it sounds fascinating. It looks very like a great possibility. But
okay,
maybe Yuyang can comment on that. But it's very, very interesting.
All right.
I can jump in and say that we're working on this. We're really hopeful that we can get some diagnosis of people that don't have genomes through the facial phenotype, and we're working with just alt-matcher people to look at this.
Excellent. Okay, I think that actually wait on. Here we go. There's I'm going to make an error in pronouncing this person's name, so apologies in advance. But Thaloka Ratnayake asks about, from a community pediatrics perspective, asking about challenges to requesting whole genome sequencing, and if basically it's not ordered under the right panel, like R29 panel, what's the likelihood of prioritizing this gene going forward? Well, I think that's a difficult question, isn't it? About is it going to get picked up if people order the wrong panel? If you looked at if people were guided purely by panel app, would this still have been picked up? I'm guessing that it probably would have been picked up as, say, as a low category variant, even if it doesn't come up in the actual prioritized panel variants. Guess it depends on how hard people actually dig. I think that's maybe one that we need to answer offline. Whatever, I suspect you're now going to result in a lot of diagnosis. Okay, so I've got a comment here. It will be green on panel app shortly. So it would not have been reported in GMS until it is green. Okay, obviously it wasn't reported in GMS because it wasn't discovered until just now, so that's not a surprise. With that, I think we've actually come to the top of the hour, and so I'd just like to thank both of our presenters. Daniel, thank you for an excellent presentation, Yuyang too, and to the two teams who've made this outstanding discovery, which really drives forward understanding of neurodevelopmental disorders, but also in the role of non-coding RNA in monogenic diseases. It's a fantastic contribution to biomedical research, and you're both groups are to be congratulated for it. I look forward to seeing everybody who's on the call on the next meeting on June the 25th, which, just to reiterate, will be Professor Raj Thacker from University of Oxford, and secondly, a presentation from Genomics England about our Cancer 2.0 long-read and multimodal programs. With that, I'll say farewell. Thank you, everybody.